But what the scaffold N50 values weren't too different from the values with the long reads, except if you dig down into the details of those scaffolds, the short read assemblies were giving thousands of errors, sometimes tens of thousands of errors in contigs or misjoins or gaps and so forth that were not present in the long read based assemblies. And with the high-C maps, as everybody know, we can get these chromosome maps that match the karyotype, and shown here for a correlation for the Anna's hummingbird. It's not only there's data that's missing, but there's also data that's extra that should not be there in these old assemblies. For instance, here, these red labeled exons is a duplicated gene that was thought to be a gene duplication for this zebrafinch, but it turns out it was the two alternative haplotypes not being properly phased in the assembly process. And we found out that this was a big deal. You must phase haplotypes in order to get a correct assembly. Otherwise, you make these mistakes for thousands of genes, 3,800 here for the zebrafinch, 600 genes that were falsely duplicated for the previous hummingbird and so forth for the platypus. And even of the missing data, it's not random. What we're finding is that the higher the GC-rich content you have, actually the closer it is to the promoter, the 5' start site of these genes, like the UTR sequence, the higher GC content you have, the more likely the short read enzymes were not getting through those sequences. But the long read technology, like PacBio and Nanopore, were getting through these GC-rich structures. And here is shown for vertebrates broadly, mammals, birds, reptiles, fish, and so on, that as you get closer and closer to the to the start site of the protein coding genes and long non-coding RNAs, you have this rapid increase in GC content that goes from 50 to 70%. And that was missing in half of the genes in many of the prior assemblies, so you're missing your promoter regions. So that's the motivation, some of that went into why we need a new pangenome reference. And around the same time, the telomere group were utilizing these advancements and generating new ones to try to generate as complete human genome assembly as possible. And make a long story short, here we have, because many people have seen the paper, the first complete human genome from chromosomes 1 down to 23, but just showing 15 here. And everything in black here, these little black tick marks and these big black bars here, is what was missing in the previous GRC 838 reference, mostly in centromeres or repetitive regions throughout the chromosomes. Mind you, the GRC H38 reference took over several $1 billion to generate and over 20 years to correct. And so what did the trick here is combining two long read technologies, PacBio Hi-Fi, which was highly accurate long reads, and Oxford Nanopore ONT reads, not as accurate, but they were used for scaffolding across gaps that was in the Hi-Fi based contigs. Then using other approaches like BioNano to support different corrections to the reference here. There was a lot of manual curation as well that went into making sure that all the gaps are filled properly. And further, as you can see here, in the coverage plot across certain regions of the genome, there was no haplotype switching. So here in red is the alternative haplotype, and in black is the haplotype that's appropriate for this particular region here. And there was a lot of this going on, as well as collapsing of sequence that needed to be expanded out. So it's not just gaps that's a problem, but it's collapsing and switching of alleles that had to be corrected as well. And so just to show some numbers here, there were 42 left unlocalized segments of DNA, 0 in this T to T reference, 127 unplaced contigs or scaffolds, there's 0 here and so forth. The CMH13 is actually a little bigger than GRC38 because it's more complete and so on. And what we found also was important was measuring the QV accuracy of the base calls, which most people don't do with genome assemblies. For the CMH13 reference, it's 70, which means no more than one error per 10 million base pairs. Okay, so with all this motivation, NIH decided to fund what we could now call a human pan-SPINOS genome reference to replace the current GRC 838 reference. And the goal here was to create high quality, near complete and near error-free genome assemblies, representing at least 95% of human genetic diversity. And that was SNP diversity, indel diversity, structural variant diversity. And through some calculations, an estimate was made that you need 350 diverse individuals, which will give you around 700 haplotypes to achieve that goal. That's a theoretical calculation for SNPs at least, based upon the 1,000 Human Genomes Project, short read data. And to finish a subset of these genomes, if not all of them, a telomere or telomere, and to be able to utilize this reference for global medicine, basic science, map all available variants and haplotypes, be an international project, even if funded here, primarily from NIH. And we are collaborating with different groups like H3 Africa, GA4, the Global Reference Alliance, and to be inclusive, not just of samples, but of participants. So diversity of people sequencing diverse genomes and of cultures as well, and sensitive to the needs and desires of different groups internationally. But to try as best to make this an open access initiative. And the goal here, if you're going to squash all these genomes together, you need a high quality pangenome reference is going to require that the genomes going into that pangenome be high quality as well and meet a minimum set of metrics. And so this is the website that several of us from the VGP and other consortiums got together, competed for the funds, we won the funds, developed this website here, with putting forth diversity and inclusion as a primary goal here. And here is the structure, which keeps evolving, but we have a steering committee. You'll recognize some of the names here, different groups from samples of consent to assembly, to generating the pangenome, to resource sharing and so forth. And the first experiment we did was to take one individual human, HG002, an Ashkenazi Jewish person, and apply all the available technologies and algorithms to this one individual available at that time in 2020, and have assembly competition, basically. We call it a bake-off. And there were 23 assemblies from 14 groups around the world submitted with all the latest different algorithms at that time using Hi5, PacBioData, Nanopore, linked reads, and so forth. And I'll just say right away, because of the sake of time, Hifiasm rose to the top in this comparison. And when I say rose to the top, we compared 60 different metrics amongst these 23 different assemblies, including the ones proposed by the VGP consortium, where we're trying to reach a metric getting a QV 50 or higher, N50 contig of 10 million base pairs or higher, as much of haplotypes phased as possible, and so on. And so I don't have time to get into all the details here. I just show you some of the highlights. Here are the 23 assemblies, where some of them are phased by haplotypes, like this VGP maternal and VGP paternal assembly, phased by haplotypes, color-coded according to their haplotype phasing, or other features. And this trio-based Hifiasm assembly, which was assembly number 9 here, rose to the top, not in all categories, but in most, like gave the highest QV values for base call accuracy, among the highest haplotype phasing blocks, among the highest contiguity for the contigs. And here for haplotype phasing, it did pretty well, where haplotype completeness was almost 100% for each haplotype separately, Some of the assemblies that didn't fully phase the haplotypes, they had merged haplotypes, the red and the blue here. And the total gigabases phased for maternal and paternal chromosomes were the most complete for this trio-based Hifiasm assembly, and had the least haplotype switched errors here, or among the least for the contig-based assemblies. And so what was so special about this type of approach from Han Li's group is that not only does it try to phase the haplotypes, but it phases the haplotypes data in the assembly graph here, as opposed to phase and assemble contigs separately. And so once with this phasing of the data using the parental data, maternal and paternal data in the assembly graph, that process itself led to a more accurate assembly in both structural accuracy and base call accuracy. And so what we did is we utilized that approach and the best of all the different scaffolding approaches that we had based upon our experiences with HG002 bake-off and the VGP project itself. And we came to an approach where that gave us the highest quality assembly, at least in early 2022, using the trio Hifiasm approach, then scaffolding with BioNano, then further scaffolding with Hi-C, filling in GATs with ONT, and then manually curating to get in the final assembly. And we found that the regions that the ONT data filled in that the Hi-Fi data didn't get correct were homopolymer regions like these T and Cs, as well as regions, if you can read the fine print here, that were GA rich. And many people knew about this already, that Hi-Fi data has difficulty, Hi-Fi technology has difficulty sequencing GA-rich repeat sequences, which there are about 20 of them in human genomes, but hundreds in fish genomes. So it does make a difference across species. And so when we look at the Hi-C plots now and compare with CMH13 for this more complete HG002 assembly, we're getting close to CMH13 completeness. Here, going from 0 to 100% completeness relative to the CMH13, you can see that most chromosomes are near 100% complete for this HG002 assembly. Some chromosomes not, like chromosome 21 here, or 13 and 15. These are the acrocentric chromosomes where their telomeres or centromeres are near the ends of the chromosomes, and they're the hardest to assemble with the rDNA arrays, which makes sense. And so here is just to show haplotype completeness of this HG002 assembly for paternal and maternal contigs. And an alignment of this HG002 to CMH13 reveals that the gray bars here, so blue is paternal, red I think is, maternal. We get almost complete alignment of HG002 against CMH13, except in these gray zones here. And it's what these gray zones here are telomeres. It's not actually that they're missing in this HG002 assemblies, is that they're so divergent between HG002 and CMH13, and also within HG002 that they don't align well. And so that was an interesting discovery in both CMH13 and HG002. By the way, I'll explain it here. I didn't put a slide, I meant to say this. Why don't we use the technology that gave us CMH13 now to do a whole bunch of humans? The reason why we can't do that is CMH13 is an effectively haploid cell line where one set of chromosomes entirely duplicated in utero while this embryo was developing in the mother's womb. One set of chromosomes in the starting germline sperm cell duplicated and became a complete copy of the other and kicked out the maternal chromosomes. And so now you totally get rid of the haplotype phasing problem. And so that prevented haplotype phasing errors. And what we need, though, is a technology that can generate diploid telomere to telomere assemblies. And we didn't have it yet at that time, and still don't actually, as it can get to the level of CMH13. So nevertheless, we got close, where here you can see the gaps in these gray triangles, like here in chromosome 1 or chromosome 8. They're roughly, on average, 4 gaps per chromosome, as opposed to hundreds in the assemblies that were being done with the short read or even the prior long read approaches. And so, so looking at the haplotype diversity here in these black lines across the 2 different haplotypes, we can see translocations of sequences from in one haplotype versus another. We can see as we get close to the centromeres in red here, we see increased SNP and indel diversity. We also see diversity in synonymous and non-synonymous mutations between the 2 different haplotypes all across the genome, and they tend to be focused on certain types of genes. I can't remember the category genes. Well, this is submitted as a bioarchive. You can go look and see that. We also see there really are up to about 60 genes missing per haplotype in these different individuals, GRC38 or HG002 and CMH13. And they're overlapping in what is missing between the 2 haplotypes, and they tend to be of genes that are already duplicated. So there's like 20 copies of the gene in one individual and 10 in the other individual. And so that's interesting biology, where there's certain categories of genes from one person to another, or even in 2 different haplotypes of the same individual that are absent or present. And they tend to be genes that are in primate-specific lineages and that function in the brain. So now the pangenome reference, with Hifiasm approach being the best approach that we have up until in 2020 to 2021, the Pangenome Reference Consortium took a set of 42 or so 1,000 Genomes Project individuals and resequenced them using PacBio data, ONT data, Hi-C, nanopore data. And here you can see the breakdown of individuals, including of different African populations, Puerto Rican and so forth, and some European. Actually, we didn't focus too much on European here. And here is a PCA plot of the genetic diversity of the individuals that went into this initial sequencing, where we created contigs. And here is a PCA, not a PCA plot, a t-SNE plot of the same type of data sets, where in red text here are the samples that were used that went into this first pangenome reference. And just looking at the trio-based Hifiasm assembly now of each haplotype, maternal and paternal, and seeing green is, we use tools to determine, is this complete and correct? And what I mean by complete, it still has gaps, but what is sequenced, is that complete and correct? And the answer is that roughly, 3 gigabases of sequence minus what's in these different colors down here, less than 10 or so a million base pairs, had some errors in them, whether they be collapsed sequences of false haplotype duplications, erroneous sequences that turn out to be mitochondria inserted into the genome assembly, or some viral sequence that doesn't belong there, or some bacteria. And so the incorrect assembled sequence is uneven across all these assemblies, and has mostly to do with level of heterozygosity or quality of the Hi-Fi data. And you can see between women, female and male genome sequences, the males all have one haplotype that is shorter than the other. The reason why is because the Y chromosome is shorter than the X chromosome. And so it's not that one is more complete than the other, it's the differences in the sex chromosomes, which means we assembled them right. Looking at indel diversity and structural diversity across these haplotypes, we can see, as an expected, African populations have greater diversity or increasing number of variants relative to the current GRC reference, more than samples from the United States or East Asian and so forth, for both structural and SNP diversity. And here is there are tools that Benedict Payton's group and Eric Garrison's group and other groups have developed that now have taken these 90 something haplotypes from these 42 individuals and created a pan-genome graph from them, where you can walk through the graph and look for structural variation like indels or SNP diversity across this graph and find the different diversity in each of these haplotypes. And they use two different approaches. One, which uses the cactus algorithm to perform a reference-free alignment where it didn't depend upon any one reference, although it did map all of them back to GRC38 or CMH13. But to make the graph, it didn't depend upon any one reference. And another that did an all against all alignment called Pangenome graph that included centromeres. The other one didn't include centromeres. And we just submitted this last week. So I encourage you to go read that. I'm just going to walk quickly through it because of the sake of time. Here is an example of what the different haplotypes look like for our HD locus, where you can walk through the assembly graph here and see different bubbles that correspond to different haplotype diversity here for these different structures of this gene family here. And to end off with, Adam Phillippe's group has taken what the lessons learned here from all these approaches and generated a new algorithm called Verco, which tries to do as what Hifiasm does, is to, in the assembly graph, separate the haplotypes where they differ in the two different haplotypes, merge them together where they're homozygous, and then walk through this assembly graph with the HiFi and the ONT data assembled together, not in two different steps. And then try to use that data to then pull apart the homozygous region here into their respective, to be with their respective haplotypes and get a more contiguous assembly. And long story short, with Verco in trio mode, they were able to get the highest contiguity of any human assembly for contigs, and also low error rates in haplotype switching and other types of error, high QV values similar to the curated version of the HG002 assembly we have here. And so second to the last slide, I'm just pulling this back out from our reference assembly that we generate for HG002. Here, I just want to show you to be careful of collapse regions. Here we have coverage plots across the centromeres of HG002. And you can see here where the coverage changes in the two different alleles. And this happens more often across the centromeres than the rest of the genome. Why? Because there's a lot of repetitive sequence here that's being collapsed. And it's hard for these algorithms to separate out the two different haplotypes when you have repetitive centromeric regions. And so this is not just me working on this project. It's a consortium of many people that I showed you earlier. And so I just want to thank them for really getting together and really putting the brain power of the world's leaders in assembly and so forth to try to do this project. And I'll stop there.
All right. Thank you, Erich. That was a good one. So we have 10, 15, maximum 15 minutes for question and answer so that we can wrap up. So if anyone have any question, please do put your question in the chat box or raise your hand, please. And Zara will take them for you. So I think maybe I can just start off. Erich, I think with Verco, is it applicable now to is it possible that we can use Verco for vertebrate species? I don't think so. I mean, what are the potential bottlenecks? So
they did try it on a few other species besides human, and it does work.
Okay.
Parameters need to be different, for different species. I wouldn't say it's exactly prime time ready yet. It's a little cumbersome to use, at least for the folks that I've spoken to. So I would give it another 3 to 4 months of development before it's useful for production for other vertebrate species or species
Okay.
in general.
All right. So what we know is that there's a chance that even for polyploid organisms, there's a chance that Verco could potentially work in those organisms, I think.
That's right.
All right, cool. I mean, that sounds good. So Zara, I think some questions are coming through. Do you want to take them?
Yes. So the first question is, how do scientists benefit from this project in terms of knowledge transfer, and training?
Mm-hmm. Yeah, so for the VGP project, and it bled into the telomere as well for the Pangenome effort, we have webinar sessions where we train people, anybody who wants to join. Well, there's some prerequisite of knowledge they must have, but they can join with students, faculty, others, to learn how to do these assembly pipelines, or webinars on how to isolate DNA and so forth for high quality DNA isolation. So that's one. The other is that these are open door consortiums. Anybody who wants to join can join, as long as they play by the rules of fair and open access of data, and making sure that people get proper credit and so forth. So that's another. And then the students and postdocs are being first authors in some of these papers that are, that has a consortium behind them. And so they're really benefiting that way as well.
Great, thank you. I'm sure that's very encouraging.
Yes, so anybody here that wants to join, just let me know.
Yeah. So Erich, are you happy to share your slide with us, please, if privacy allows?
Yes.
All right, so maybe, I think you just send it to, I'll just maybe do a follow-up, or you just send it to my email, whichever one is.
Okay.
Okay. All right, so we can share it with the community. Okay, Zara, over to you.
Yeah, okay, Yasmina, please go ahead.
You're on mute.
Yeah, I think she's trying to unmute herself. Yasmina, can you hear us?
You have to unmute Yasmina.
There she goes. No, you just muted back.
Yasmina, you have to unmute. Okay.
There you go.
I was trying so hard to unmute myself, okay, because you muted me. Anyway, thank you for the great presentation, Eric. I'm just wondering, I mean, what are the moves now? How do we scrap CRH38, or are people now moving towards using CHM13? Just in our group, I'm in the same group as Sean, so we did quite some analysis using HT38. So what do we do now?
So yeah, this is something that we're even trying to figure it ourselves, and trying to engage both the basic science and the clinical or translational research community, because so many scientists and companies have built tools that cost tens of thousands to millions of dollars on GRC838 reference. And it's not easy to change over to a new reference. Yes, if you want your science to be more accurate, we're encouraging you to use CMH13 for now, or this Pangenome reference. NIH is trying to, or they're not trying, they are working on tools to make this reference more easily usable in the different databases. And you're going to have to think about using references in two ways. One is that there's one individual reference, like CMH13 or HG002, that's complete or almost complete. And then there's the Pangenome reference. And that's going to be more complex, because you have this complicated graph, eventually through 700 different haplotypes or more, that's going to be hard to figure out, which particular individual or population you're looking at. And so new computational tools need to be developed in order for the community to be able to utilize a Pangenome reference. And the team some of the people are working on those new computational tools, but they're not enough people. we need more people to join that effort to make these pangenome references more usable.
All right, thank you, Erich. So I think another question I have is again leading up to, I think from Daniel's point, so which relates to the Human Pangenome Project, because in the project you have, for instance, if it's like imagine, you have a single reference genome, and then you have, looking at diversity. You have the benefit of being in both worlds, the biodiversity and also the human side of things. So what has been your experience so far, especially with the VGP, where, and again, you can also link this to the Human Pangenome Project. Do you kind of start off? Because one thing I think is that it's always good to perhaps, again, depending on resources, look at the representations at maybe the tree level, the eukaryotic tree level or family or order, and then potentially expand into geographical locations. And so what do you think, and how does that connect again with the Human Pangenome as well?
I think it's better if someone forces you to make a choice, between most species of a continent or of a particular lineage versus population data from one species, from fewer species, I would rather go with most species, because there's a lot of genetic diversity in the animal world, in the plant world, and so forth, that we could learn from, that we're not going to learn, that even having one individual of that population is going to be valuable to learn about that genetic diversity. A lot of these species are on the verge of extinction. There won't be a population to screen from in the future. And so I think for conservation reasons, it's also good. I think also to do population analysis, it's more expensive if you want to do it high quality like we're doing here with long reads. And so 350 individuals versus 350 different species. I think you get more value out of 350 different species when you spending that much money. So in time, this technology is going to get cheaper and cheaper. Third generation is going to get cheaper. And then it's going to get cheaper to do population analysis in the future. So yeah, if you have the money and the will and the need and the way, like for a human, for a few other species, Arabidopsis or, corn plants or something like that, yes, then do a pangenome. But if until then, until it gets cheaper and faster, I would focus more on species.
Yeah, absolutely. I thought so as well. And also one of the experiences again is that, with the pangenome, especially at the biodiversity level, you have a single individual. If they cut across multiple countries, then that is multiple permits and
Right.
ABS. So again, and all of those, so even trying to, sort it out for one country is a huge challenge. And then I think I do agree with you that the focus would be on the, the genetic diversity side of things and multiple species. Okay, Zara, do you have any question, or does anyone have any question? Any other question? We need to wrap up in 5 minutes.
Yeah, I don't have any questions, and I see multiple comments, but they are to thank Erich and to congratulate him on his work. But I don't see any questions in the chat.
I'll just end off with saying that one of our biggest challenges is getting consent across different continents and having the same principles applied. Some countries, have policies that genomes from country X, Y, or Z cannot be public, right? Or you need special permits of humans, that is, And so this is a challenge for us in terms of because we want this to be open access. We're making cell lines, most of the samples that we're using as well, which causes some permit, or IRB issues. So there's a public policy aspect of this that's different than working with non-human animals that I'm learning about, if you really want to create a pangenome reference that represents global human genetic diversity.
Yeah, absolutely. I do agree. Okay, so any more questions, comments, suggestions? All right, so Zara, over to you now.
Yes, So if we don't have any more questions for today, I'd like to thank Professor Jarvis for his very nice presentation. I'd like to thank everybody who attended the meeting today and for the interesting discussions we've had. So we'll meet again in 1 month's time for our monthly